New compound shows promise in reversing Alzheimer's symptoms in rats. A newly discovered chemical compound has demonstrated the potential to reverse Alzheimer's symptoms in animal trials, offering a glimmer of hope in the ongoing search for effective treatments. Researchers are now eager to advance to human clinical trials following promising results in a rat model of the disease. The challenge of Alzheimer's and beta-amyloid plaques. Alzheimer's disease is characterized by progressive cognitive decline, often linked to the accumulation of beta-amyloid plaques in the brain. While the precise role of these plaques remains debated, whether they cause the disease or are merely a symptom, they remain a central target for therapeutic interventions. Current Alzheimer's treatments primarily focus on managing symptoms rather than addressing the underlying causes. This has driven intense research into drugs that could tackle the disease at its roots. How the new compound works. Copper regulation. The new compound works by selectively removing excess copper from beta-amyloid plaques. Copper ions have been identified as contributors to the aggregation of these plaques, with imbalances in copper homeostasis potentially exacerbating the disease process. Biochemist Giselle Cerqueiro from Federal University of the ABC, UFABC, in Brazil explains, studies have shown that genetic mutations and enzyme changes affecting copper transport can lead to its buildup in the brain, promoting plaque formation. However, it's important to note that some Alzheimer's patients actually experience copper deficiencies. The compound is designed for those with excess copper. From virtual screening to animal trials, the research team first screened 9 compounds, 8 imines and 1 quinoline-based, using computer simulations. The simulations identified 3 candidates, L09, L10, and L11, that could potentially cross the blood-brain barrier and be administered as pills. Lab tests revealed that L11 was toxic to brain cells, worsening oxidative stress. In contrast, L09 and L10 showed low toxicity and protected cells from damage. Compound L10 then stood out during trials in rats engineered to develop Alzheimer's-like symptoms. L10 restored normal copper levels in the hippocampus, the brain's memory center, reduced neuroinflammation, and improved spatial memory in maze tests. L09 and L11 had weaker effects. The path forward, clinical trials and potential impact. The team is now preparing for human clinical trials, which will determine if the compound's effectiveness translates to patients. With an estimated 55 million people worldwide living with Alzheimer's, even a partial improvement could be significant. Cerqueiro emphasizes that the compound is simple, safe, and effective, and potentially more affordable than existing drugs. Even if it only works for a subset of patients, given the multiple causes of Alzheimer's, it would still be a major step forward. The development of this compound represents a promising avenue for Alzheimer's treatment, though further research is crucial to confirm its efficacy and safety in humans.